understand what an immune response is, we must understand the important cellular components of the immune system. So the cells of the immune system are comprised of white blood cells, which can be further categorized into lymphocytes and leukocytes. So we'll first start with leukocytes. Leukocytes are the disease-fighting white blood cells, so they function to identify and eliminate invaders by releasing destructive material to kill them or eat them up. So I like to consider these cells the cleanup crew of the immune system. Then we have lymphocytes. So there's B cells, which contain antibodies, and these antibodies will allow the B cell to bind to any of that foreign invader and mark it for destruction. Then we also have T cells. So the T cells help coordinate the immune response and can directly attack the invader and tell the B cells to produce more antibodies, which marks the invader for destruction. One important note here is that both B cells and T cells produce memory cells, which stick around just in case the body gets reinfected with the same invader, and in that case, the cells know exactly what to do. So now we'll get into the breakdown of the immune response now that we know all the components that it contains. So first, we'll be hopefully not infected with COVID-19, and then our B cell will recognize this foreign invader and bind to it. Next, the B cell is quote unquote activated and produces antibodies that are specific to COVID-19. Next, the antibodies will bind to COVID-19, and this will mark them for destruction. And they will be destroyed by the cleanup crew that I mentioned before and the T cells. Lastly, COVID-19 is destroyed, and in this process of the immune response, those memory cells were produced. So if the body is reinfected with COVID-19, the immune system knows exactly what to do. So it's important to understand that we have two types of immunity, adaptive and innate immunity. So let's first start with innate immunity. This type of immunity you're born with, so it acts quickly upon the presence of that foreign invader, but is non-specific. So it isn't enough to amount an immune response. It's just the beginning. And then we have adaptive immunity. So this type of immunity develops throughout life. Once you've been exposed to a foreign invader, all these cells will remember this encounter, and if reinfected, the body can mount a faster and more specific immune response against that invader. So this is important because this is the kind of immunity that vaccines will give us. Now I'm going to go into traditional vaccine classifications. So first, we have the live attenuated virus. So this is a weakened version of the disease-causing virus. This replicates in the body and stimulates the immune response, although not strong enough to cause a disease. An example of a live attenuated virus vaccine is a rubella, varicella, and mumps vaccine. Next, scientists have come up with the inactivated/killed vaccine. So that consists of virus bacteria particles that have lost disease-producing capacity. So essentially, they introduce these dead cells into the body, and the immune system can produce an immune response against those dead antigens. So examples of those kind of vaccines are polio, Hep A, and rabies vaccines. And then lastly, there's such thing as recombinant vaccines. So this is a type of genetic engineering technology where a segment of the viral genome is inserted into yeast cells, and these yeast cells will reproduce a portion of that virus. And when it grows, the body will recognize this portion of the virus and make an immune response against it. So the examples of this type of vaccine are Hep B and HPV vaccines. So in terms of the COVID-19 vaccine, the ones that are currently approved in Canada are mRNA vaccines, but we need to first understand what mRNA is. So as we all know, DNA encodes all the genetic material. It contains our genetic blueprint. And then DNA is copied into RNA, and that RNA template is used to make all functional proteins in the body. Okay, so how will this mRNA vaccine work then? So basically, as I mentioned previously, it only takes a portion of the virus to elicit an immune response in the body. Scientists chose to use the SARS-CoV-2 spike protein portion of the virus, and this portion of the virus is critical because it's how it enters our cells. So COVID, as long as Dewey, has DNA that codes for all of its parts. So scientists took that DNA and identified the specific region that codes for only the spike protein and made it into RNA. And that RNA is what's being delivered to us through the vaccine. So pretty much our cells receive this RNA and interpret this as a set of instructions to make that spike protein. But once your cells have made this protein, your immune system is like, wait, we've never seen this before. basically goes into attack mode. And then the cascade of events that I highlighted before, called the immune response, will produce the right cells to equip our body with immunity. Note that since we're only being delivered the spike protein portion of the virus, it's not strong enough to actually give us the full COVID-19 disease. So now let's get into the composition of the COVID-19 vaccine. So first we have mRNA. So as I mentioned before, that encodes the viral spike protein, so this SARS-CoV-2. Then we have lipids. So lipids are important because they ensure that the mRNA enters the cell successfully. So there'll be a lipid nanoparticle layer that surrounds the mRNA. Then we have sugars. Sugars are critical in ensuring and functioning that the vaccine is protected and stabilized during freezing. And lastly, we have salts. So salts allow the cell to retain proper shape and environment and helps the body absorb the solution. Here I highlight the specific ingredients in both the Pfizer and Moderna vaccine. They're very, very similar. So they're both mRNA vaccines. They're both 2 doses. They differ in the amount of vaccine that's given in each dose. So on the left, we can see Pfizer has 30 micrograms of vaccine in each dose, and Moderna has 100 micrograms of vaccine in each dose. Their ingredients vary very minorly, but they both contain salt, sugars, and lipids. I want to direct your attention to the middle of this slide. So as I said before, they're both an mRNA vaccine, and they both code for the SARS-CoV-2 spike protein. As I mentioned before, they're both 2 doses, and they both produced 95% efficacy. So why it's important that we take the COVID-19 vaccine? It's important because we need to achieve herd immunity. So herd immunity is when most of a population is immune to an infectious disease, and this will provide indirect protection to those who are not immune to this disease. As a result, not every single person needs to be vaccinated to be protected, which helps ensure that vulnerable groups who can't get vaccinated are kept safe. So in order to achieve herd immunity and stop the spread of the disease, experts have estimated that nearly 60 to 70% of the population must be immune to COVID-19. So that means it's very much a group effort. Here I highlight the difference between natural immunity versus vaccine-induced immunity. So a common question is both natural COVID infection and a vaccination are expected to protect you from getting sick with COVID-19 in the future, but how are they different? So I've highlighted 2 major things. So the safety in natural immunity is worse because you're more likely to be hospitalized. So it was actually reported that not just the older generation is hospitalized, up to 12% of hospitalizations in Canada were under the age of 40. Further, the immune response from the natural COVID-19 infection could be deleterious. So the virus itself is better at dysregulating the immune system because it's a live virus and it's not just a component of it. Versus vaccine-induced immunity, so adverse events have been extremely rare. There have been severe allergic reactions reported, but nothing further than that. And the immune response is regulated as the virus isn't alive and can't evade or outsmart the host immune system. Secondly, we want to look at protection. So we don't know currently how long natural immunity lasts. So we don't know whether you can be, it is possible that you can be reinfected with COVID-19. And that's why you're still, advised to get the vaccine, even if you've had the virus. And whereas the vaccine-induced immunity, we know that it offers long-term protection. The last component of my presentation is debunking COVID-19 vaccine myths, just because there's a lot of vaccine hesitancy going around, and just to clear anything up. So the first case is, was it produced too quickly that efficacy was compromised? So I put this little graphic of the woman here saying, don't vaccines take a decade to develop? And yes, in the past, they've taken this long to develop, but here's why COVID-19 vaccine is safe. So the first reason is major collaborations. So pharmaceutical companies and government bodies invested a significant amount of resources into quickly developing a vaccine for COVID-19 because of the worldwide impact of the pandemic. Secondly, no corners were cut in the production. So in order to FDA approve a drug, there has to be a randomized placebo-controlled phase 3 clinical trial that has to be complete. And this has been for both Moderna and Pfizer. Research is not starting from ground zero. So the family of coronaviruses has been widely studied before, so it's not completely new to us. And lastly, mRNA vaccines are much quicker to develop than other types. So RNA can be readily produced in the lab from DNA templates versus if you're like inoculating a virus in a chicken egg, it takes a while to inoculate. Vaccine hesitancy case number 2 is can the vaccine cause infertility? So why did this come about? It was seen or said that the spike protein of SARS-CoV-2 was structurally similar to syncytin-1, which is an important protein in the development of the placenta. Skeptics are worried that the vaccine will target this protein as well and render females infertile for an quote-unquote unspecified duration. This is not the truth. So while the coronavirus spike protein and syncytin-1 do share a small string of amino acids, which is just proteins in their structure, the similarity is too small to warrant an immunologic response. And during natural infection, the immune system generates the same antibodies to the spike protein that COVID-19 vaccines would. So if COVID-19 affected fertility, there would have already been an increase in miscarriage rates in pregnant women infected with the virus. Case number 3, can the vaccine cause severe allergic reactions? So as with all vaccines, there is a chance that there will be a serious side effect, such as an allergic reaction, but these are extremely rare. The FDA has stated that only people who have previously had severe allergic reactions to vaccines or specific ingredients in the vaccine should avoid getting the shot. The CDC has reported that safeguards are in place at vaccine locations. So you have to stay for a certain amount of time. If you are allergic, you have to bring your EpiPen and you are monitored on site. But it is recommended to consult your physician if you have any reason to question the administration of the vaccine. And lastly, can the COVID-19 alter my DNA? So this is no, the answer to this is no. The DNA is stored in the nucleus of the cell, and this compartment of the cell is extremely well protected. Not much can get in there. So mRNA vaccines do not enter the nucleus, and therefore they cannot interfere with your DNA at all. So mRNA will stay in the blue part of the cell here that I've highlighted. And in fact, as soon as the cells make that spike protein, the mRNA transcript that was initially delivered through the vaccine is degraded immediately, and all that you end up with is an immune response against COVID-19 with those antibodies that are left. So that concludes my presentation. Thank you for giving me the platform to do this. This was awesome. So I will pass it over to Rajiv now. Okay.
All right. Thanks, Alex. That was excellent presentation. And so I'm going to build on that one and present it today. So hello, everybody. My name is Rajiv Nepal, and I am a medical advisor for COVID-19 vaccine, work with Pfizer. And I'm very delighted to be here to be presenting our data. So let me share my screen. So as I said, some of the stuff that I'll be talking about would be a repetition. So maybe that will help to make some of these complicated concepts a little bit clearer. So here's a general disclaimer. Just want to point out that this vaccine, COVID-19 vaccine, is authorized to use in Canada under an interim order. And like all other vaccine, it comes with warnings and precautions. And as Alex said, it's contraindicated in those people who are allergic to the active substance or to any ingredient in the formulation. So here's my presentation outline today. I will start with a few introductory slides, and they kind of, as I said earlier, builds on to what Alex just presented. And then I will get into the vaccine clinical development program. So here's the Pfizer-BioNTech COVID-19 vaccine. This is indicated for the prevention of coronavirus disease 2019 called COVID-19 that is caused by the virus SARS-CoV-2 in individuals 16 years of age and older. The vaccine comes in, the dose is 30 microgram, and 2 doses are given 21 days apart. And it comes in a multi-dose vial, 6 doses in a vial after the addition of diluent. And it's frozen, stored in -80 freezer before the use. It's also stable for 5 days at 2 to 8 degrees Celsius. So in a normal freeze. So I would like to start this presentation with this slide by explaining how SARS-CoV-2 virus infect human cells. I like to start with this because this also helps understand how our mRNA vaccine actually works. So as Alex said, the virus needs something to hold on to before they actually enter the cell and establish an infection. So every virus is unique in its own way and uses some certain kinds of molecules on our cell surface to gain that access into the cells. So our cells has plethora of these molecules there, and they have all their physiological functions, and virus just finds one of those molecules and exploits it to enter. So the molecule that virus uses for entry into human cells is called ACE2. So you don't want to go into detail, but this is a molecule ACE2, and the protein that's on virus surface is called a spike protein. So these spikes are the one that's sticking out of the virus, and that's how, by the way, they get the name coronavirus. So once the virus is inside, what it does is it releases its viral RNA. So usually in most of the cases, the genomic content is stored as a DNA, but there are some viruses where instead of DNA, they have RNA as their genome. So SARS-CoV-2 is one of those viruses. The RNA is the genome, so that genome is now released into the cytoplasm. So at this point, the virus now takes control of our internal cellular machinery. So what that means is now this RNA, which has got codes for different viral proteins, will be, so our cellular system, our cellular machinery will start making viral proteins at that point. And some of those proteins are the structural protein of the virus. Some of those proteins are used to make the further copies of the viral genomic RNA. So in essence, what virus does here is takes control of the cellular machinery and manufactures its parts. And at some point in the manufacturing, the assembly starts, and all these different virus parts, they come together and make a virus. So something, you start with one virus in a simplistic sense, now turns into thousands and thousands of virus inside the cell. And then they are eventually released and they repeat this cycle of infection. During this process, they expose themselves to our immune system. And as Alex said earlier, our immune system then recognize these virus proteins as foreign and start eliciting immune response. So I wanted to present this first because our vaccine, in some ways, works like this, except that the vaccine will elicit the strong immune response without producing the disease. So that's the main difference between vaccine and the natural infection. So the protein that's on virus surface, spike protein, that's the one that holds on to our molecule ACE2 to gain entry into the cells. And a lot of work has been done on this spike protein. Remember, we had two major outbreaks of different magnitude in past from the coronaviruses. One was early 2000 SARS, and another is MERS in 2010s. And a lot of work has been done. And what scientists found out was if there's an antibody that binds to this spike protein, that will prevent the entry of this virus into the human cells. So based on all those work, we chose spike protein as an antigen in our vaccine. So now let's talk how the vaccine works. So we have deep insight into the mechanistic detail of how this vaccine works. So in this case, the vaccine is mRNA vaccine. And as Alex said, this vaccine now has the codes for the spike protein. The mRNA is a very fragile molecule. So to protect the mRNA, a lipid coat is applied, a coat of lipid is applied, so a fat layer is applied, and this is what you call a lipid nanoparticle. So you can think these lipid nanoparticles as vehicles for this mRNA to protect them. And once you inject an individual, these LNPs are then taken up by human cells. Here, the green one is the human cells. And once it's inside, just like the virus, that mRNA is released. And now that mRNA will it contains the genetic codes for the virus spike proteins. So what our machinery does at that point is it starts manufacturing those virus proteins. So just mind you, here we are just providing the cell a kind of a blueprint, a kind of an instruction to produce one part of the virus, not the complete virus, just one small, tiny part of the virus. But once these proteins are synthesized inside the cells, our immune system recognizes them as foreign and mounts an attack. And while doing so, they activate different kinds of cells, including T cells and B cells. Some of those T cells, they provide help to B cell to make high quality, neutralizing, long-lasting antibodies. And these T cells are called helper T cells. There are some other T cells which our immune system trains and creates a kind of a fighter T cells, killer T cells. They are trained in one-to-one kind of a combat, and they are trained to recognize and destroy virus-infected cells, human cells. So this platform is able to elicit both T and B cell response. And during the process, they also create memory T and B cells. And as Alex said earlier, these are the cells which remember the earlier infection and are responsible for providing that long-lasting protection. So we all are aware that we were able to develop this vaccine in a relatively short period of time. But if you look into the vaccine development process, it has followed the normal vaccine development principles. And these are the three principles I've listed here. The first one is the vaccine must be proven effective, meaning it has to prevent COVID-19 in at least a majority of the vaccinated people. The vaccine must be proven safe and with robust safety data generated from thousands of people. And the vaccine must be consistently manufactured at the highest quality standards. So during the process, we were able to do it in a relatively short period of time while still ensuring highest compliance, quality standards, and safety. And there are many reasons that contributed to it. A few of them Alex just mentioned. One is a lot of work in terms of antigen selection was already done based on our experience with SARS and MERS. Even the platform, which is called cutting edge, very new platform, mRNA vaccine platform, if you ask scientists, it's not really new. Scientists have been studying this platform for decades. It's just the first time it's being commercialized as a vaccine. And then there was this unprecedented collaboration between different entities, including but not limited to government, academia, industry, biotech, regulatory bodies, and also most importantly, the participants of all these different clinical studies. So with that background, now I would like to get into our clinical development program. So it started as a phase 1-2 trial, and those studies were carried out in Germany and the US. And at that point, we were toying with a few different spike protein antigen forms. And we are also experimenting with a few different flavors of mRNA platform. And based on the results that we obtained from our phase 1-2 study, we selected this candidate called BNT162b2 to progress into phase 2-3 of the clinical study. So in the interest of time, I'm just gonna get into the phase 2-3 part of this study. This was a randomized, placebo-controlled, blinded study. This started in July 27, 2020, and the target was 44,000 participants. So huge number of participants in this study. As of January 2021, we've already recruited more than 46,000 participants. Mind you, this trial is still ongoing. And this trial now recruits 12 years of age and older in it. When we submitted the data for regulatory approval and for publication, the data cutoff was November 14, and at that point we submitted data for 16 years of age and older. But now we have completed the recruitment of 12 to 15 years of age and older. But the data that I will be presenting today are from our publication that came out in New England Journal of Medicine in December, and that shows the data 16 years of age and older. So one of the objectives of this trial right from the beginning was to recruit those people who are the most vulnerable, who would potentially benefit the most from this vaccine. So that involves elderly individuals and also people with chronic diseases. And we also wanted to make sure that this trial includes minorities and is balanced in terms of racial and ethnicity profile. One of the populations that was excluded in this trial was immunocompromised. And that's, I think, what is quite normal in vaccine studies, because this is a special population. So usually you want to start your trial with the general population. And once you establish that efficacy in that general population, then you move on to these special populations like immunocompromised or pregnant women or breastfeeding women. So here's the demographic characteristics of all those participants. So as I said, this trial had two arms, vaccine arm and placebo arm. And there are a little over 43,000 participants altogether here. And if you look into these two arms, they are fairly balanced. And you can also look into different subpopulations based on sex, race, ethnicity, and age, and they are all proportionately represented. Also want to point out that 65 years of 65 years of age and older represent around 21% of the participants. So with that background in terms of our trial design, let me talk a little bit about our efficacy. So I would like to start with describing how we went about our establishing efficacy of this vaccine. So just a gentle reminder, 2 doses, given 21 doses apart. And right after they received their first dose, the participants were kept under active surveillance for COVID-19. So what that means is if they developed any of these symptoms, that would trigger either an in-person visit or a telehealth, and then the nasal swab will be collected and the PCR is done. So if they show one of these symptoms and are positive for SARS-CoV-2 virus, then that would be a case of COVID-19. Now, as you are all aware, vaccine usually takes some time to show its full effect because it has to first activate the immune response, and then this immune cells start producing the antibodies. So there is some time in between. So for this trial, we counted the cases for the purpose of establishing efficacy 7 days after the second dose. So that's something important to understand, 7 days after second dose. So this trial had 2 primary endpoints. So there were 2 main objectives of this trial in terms of the efficacy. The first one was to establish efficacy in 16 years of age and older in participants who had no prior SARS-CoV-2 infection or no ongoing infection. Okay, so this is quite important because if you think of it, this is the population that is the most vulnerable to future infection. So in this case, as I said before, they were given the vaccines and they went about their daily life, but they were kept under active surveillance. And when they developed a case, those cases were counted in this case to establish the efficacy 7 days after second dose. When the cases were counted, there were 8 cases in the vaccine arm and 162 cases in the placebo arm, and vaccine efficacy is 95% with a confidence interval, as you can see there, between 90 and 98. So what that means is the true vaccine efficacy value falls somewhere in between these 2 numbers. So quite very encouraging, very strong, high efficacy with this vaccine. So that was our first primary endpoint. Our second primary endpoint was to establish, to investigate efficacy in participants, irrespective of whether they had prior SARS-CoV-2 infection or not. So the same thing is done. In this case, there were 9 cases in vaccine arm and 169 cases in placebo, and the vaccine efficacy is very similar to what we've seen before. So again, very strong vaccine efficacy in 16 years of age and older. We also wanted to investigate the efficacy against severe COVID-19. Now, when we talk about severe form of the disease, different institutions may have different kinds of criteria. For this one, there was a criteria defined by FDA. The criteria, as you can see on the slide, these SARS-CoV-2 positive patients, if they exhibit any one of these severe symptoms, admission to ICU, or clinical signs that indicates some severe systemic illness like increased heart rate, and they have a threshold there, increased respiratory rate, oxygen saturation, evidence of shock, significant acute renal, hepatic, or neurological dysfunction, respiratory failure, or death. So based on this, when we counted the case 7 days after second dose, there were very few cases. I think there were only 4 cases. So didn't really, wasn't really enough to make any meaningful conclusion. So then in this case, the cases were counted right after the administration of first dose. And when we did that, there was 1 case in vaccine arm and 9 cases in placebo arm, and the vaccine efficacy is around 90%. And when you look into the cases, the cases in the placebo arm had more severe outcomes than what the 1% showed in the vaccine arm. So again, this indicates that the vaccine is likely to be protective against severe form of COVID-19. So here I would like to just summarize this section. So again, once again, this trial met both primary efficacy endpoints. So the vaccine was able to prevent diseases in 16 years of age and older. So the vaccine efficacy was calculated to be 95%. And I didn't show the data here, but we also looked into the vaccine efficacy among different subgroups defined by age, sex, race, ethnicity, obesity, presence of different coexisting comorbidity condition like malignancy or respiratory conditions or cardiovascular conditions. And we found that the efficacy observed was very consistent with what we have observed with the overall population. So the only caveat here is this trial was not designed, it was not powered to definitely assess efficacy by these different subgroups, but it's still, nonetheless, it was important to see those results. And as I said before, this vaccine will likely provide protection against severe forms of COVID-19. So this data that I presented today is based on the cutoff that was done in mid-November, and a lot has happened since then. And as you are aware, many countries in the world have authorized the vaccine for use, and they have used these millions of doses of this vaccine in their mass vaccination campaigns. And as a result, now we can actually look at the data, real world vaccine effectiveness data from some of those countries. And just yesterday, Israel, some data from Israel were published in New England Journal of Medicine. And I'm not showing that data, but it was very encouraging to see that the efficacy that we observed here in the clinical trial is greatly aligned with what we are seeing in the real world. So now I would like to get into the safety part of this trial. So here is a diagram that tells you a little bit about the number of people that were part of this safety database. So anybody who got the vaccine or who got any of these doses will be a part of the safety evaluation, and that number is a little over 43,000. There were a little over 37,000 participants with median 2-month post-dose 2 follow-up, and this is what was mandated by FDA at that point. There were a little over 19,000 participants who had at least 2 months of post-dose 2 follow-up, and there was another subset of little over 8,000 participants, which we call reactogenicity subset. And I'll show you the data from that group in the next slide. This is the group where after giving the vaccine, you ask them for 7 days about their side effects, the reactogenicity events, and then those are presented I will present in next couple of slides. So this is how we went about evaluating safety in this trial. Just a reminder again, 2 doses given 21 days apart. And like efficacy, right after they receive their first dose, they will be evaluated for safety. And this will go on for 2 years post second dose. The first subset is, as I said, the reactogenicity subset, a little over 8,000. And basically, they were given this electronic diary and they were asked about the side effects every day for 7 days. And they filled out those replies in the electronic diary, and the data is compiled as the side effects. We also collected adverse events from all participants for 1 month, and these adverse events are unsolicited. Okay, so this is slightly different from the reactogenicity subset where we ask them, but in case of this unsolicited, if something happened, they report, and those were collected for 1 month. Any serious adverse events will be collected for 6 months post last dose, and death and related serious adverse events will be collected until 2 years after the last dose. So I'll show the data from each of these different cohorts. I'll start with the reactogenicity subset. So this side effect, you can divide into 2, local and systemic. Local is, as the name says, pain at the injection site, redness and swelling. And in this case, majority of these people, they had pain. So side effect is, again, I want to say that this is expected from this vaccine, and the majority of the people had some kind of pain, but the pain was mainly mild to moderate, and it resolved within a day or 2. Not a whole lot of participants reported redness and swelling. Now, systemic reactogenicity events, they are categorized into several different symptoms like fever, fatigue, headache, chills, vomiting, diarrhea, muscle pain, and joint pain. And interestingly, and maybe this makes sense, the older people actually fare slightly better than the younger 16 to 54 years of age cohort in terms of the side effects. And the side effects were more common and severe after the second dose than after the first dose. And if you look into the most common, which is distilled in this table here on your right, you can see all those. And even among them, the most commonly reported side effects were fatigue and headache. But interestingly, a good chunk of placebo also reported them. And there's this uncommon side effect where you see enlargement of lymph node, and that happened, it may affect 1 in 100 people, and it indicates a little strong immune response. So that's about the reactogenicity events in short. I'm just showing you the data after the second dose because that's the one little more severe than after first one. So you have 16 to 55 and 55 plus stacked next to each other. And as you can see, the older ones fare slightly better than the younger one. Around 10 to 15% have fever, and fatigue, headache, muscle pains are the most common one. And you can see there a good percentage of the placebo also report that. And majority of these events are, if you look into the color coded, they are either mild or moderate, and some severe. Severe means that these are the one that actually interfered with your day-to-day activity. And the grade 4 is something that required ER visit or hospitalization. So most of those side effects, as you can see, are mild and moderate. So here we have listed all those systemic side effects on different days. And as you can see, day 1 is when they got vaccinated. So the top one is 16 to 55, the bottom is 55 plus. So day 1, they get vaccinated, and the majority of the symptoms, they appear on day 2 and start subsiding by day 3 and largely gone by day 4. If you look into something rather objective, let's say fever, which is the most leftmost bar here, you can see that it appears on day 2 and it's largely gone by day 3. So that's the reactogenicity events. That's the side effects that we solicited. Now let's look at the adverse events unsolicited. So when we looked into those, majority of those side effects reported belong to something that we've seen with our reactogenicity events. So they were, pain, fatigue, headache, chill, that kind of stuff. So when that was taken out and vaccine and placebo arms were compared based on the symptoms put together in different organ class, you put a bunch of different similar system into one organ class, like digestive or neurological, and then there was not much different except for one condition called general disorders and administration site conditions. What that basically means is, again, administration site, injection site pains and redness and that kind of stuff. Serious adverse events, again, remember, this is the one that will be collected for 6 months post last dose. And again, we didn't see any difference between these two arms. So far, there is no clinical safety signal, serious safety signal, nothing reported based on this trial. But as I said before, this vaccine is now used in many different countries, and millions of doses are already administered. And all jurisdictions have some sort of post-marketing surveillance to look at the safety signals, to see even those rare and very rare events that we may not be able to uncover during the clinical trial, where we have only looked at 40,000-50,000 participants. So US has set up a very comprehensive, very impressive, and probably the most comprehensive in history in terms of the surveillance, both active surveillance and passive surveillance. And they regularly publish those reports, and there is nothing red flag there. Likewise, Health Canada also has its own active and passive surveillance, and it publishes those data on its website, and nothing of serious concern there. So from safety, it's very favorable and acceptable. So with that, I would just like to summarize this part. So the tolerability and safety profile of Pfizer-BioNTech COVID-19 vaccine is favorable. There is no clinically significant safety findings other than mild or moderate side effects. And the side effects were a little more common in the younger and the older population fared slightly better. The systemic reactogenicity, they were more common and severe after the second dose than after the first dose. And as I showed in one of those slides, those side effects were transient in nature, and they greatly resolved within a couple of days after onset. And as I said in my presentation, this monitoring of these clinical trial participants will continue for 2 years after the administration of their last dose. So with that, I would like to conclude my presentation, and I would like to thank you for your attention, and also like to thank the organizers for giving me this opportunity. Thank you. You're mute.
There we go. Thank you so much, Rajiv. That was an incredibly informative presentation. And also, Alex, I hope everybody's walking away. I know it's a lot to absorb at this one point in time, but we will have a recording of this webinar accessible to the registrants. At this point in time, I would like to welcome Dr. Scott Halperin and Dr. Mark Loeb. They'll be here to answer the questions that were sent in advance, starting off, and then they'll answer the questions, and Rajiv, the questions will be directed to all panelists. Dr. Scott Halperin is a professor of pediatrics and microbiology and immunology at Dalhousie University in Halifax, Nova Scotia. As director of the Canadian Centre for Vaccinology and nominated principal investigator of the Canadian Immunization Research Network, He's played a foundational role in the establishment of these and other Canadian collaborative research networks, undertaking evaluative vaccine research that informs public health policy and practice. Dr. Halperin's area of expertise is pertussis, and his research focuses on the diagnosis, treatment, and prevention of pertussis and other vaccine-preventable diseases. He currently serves on the National COVID-19 Immunity Task Force. So welcome, Dr. Halperin. And then Dr. Mark Loeb, who's also with us, is a professor in the Departments of Pathology and Molecular Medicine and Health Research, Evidence and Impact, formerly Clinical Epidemiology and Biostatistics at McMaster University, where he holds the Michael DeGroot Chair in Infectious Diseases. He's been conducting randomized controlled trials of influenza vaccine for the past 15 years and has received funding as a principal investigator from NIH, CIHR, MRC UK, and WHO for research on influenza vaccination, and is now working on COVID-19 trials. So thank you for joining us today, Dr. Halperin and Dr. Loeb. And I will move this over to Alex to start with the questions that were provided to us in advance.
Hi, Dr. Loeb and Dr. Halperin. It's great to have you guys. So a lot of these questions are directed or MPS focused. So I'll start with the first two. I don't know if you guys want to alternate between who answers them, or I guess just jump at it who speaks first. So the first question, the first two questions are, do MPS affected patients have to take the same dose as all others? And where are MPS affected patients on the list of priority in terms of Canadian vaccine rollout?
You want me to start, Mark?
Yeah, why don't you start with that one, Scott?
Sure. So the dose recommendations, first to say that for the vaccines that are available, there is very little information about people with any underlying conditions because those people were not part of the clinical trials. Having said that, there's no expectation that the immune response of people with these underlying conditions would be any different from the general population. And the dosage that would be used in the vaccines would be the same dosage. Right now, for this group of conditions, most jurisdictions will have the individuals immunized with their same age cohort. So they would not be a specific targeted population, but rather would be scheduled to be immunized when that age group gets immunized.
Okay, so they aren't classified under underlying medical conditions, or?
And again, this will vary by province because again, the rollouts are not a national thing. The rollouts will be done by each jurisdiction. So each province will have differently. So depending on where your audience is, they would have to check in their province. So my province, I'm from Nova Scotia, they would be included as part of the by age related and not a specific because their risk is not considered to be higher than the general population.
Okay, great. So the next question is a question from the EU, from Lithuania, I believe. So quote, we've approved the Pfizer, Moderna, and AstraZeneca vaccine in the EU. Based on the data, which do you believe is the safest for MPS patients who have pulmonary, cardiovascular, and allergic complications? Unquote.
Well, I can address that one. The fact of the matter is, best of my knowledge, none of the trials included MPS patients, right? So everything, and I guess my comments are the same thing as Dr. Halperin, is that we're going with sort of indirect evidence, right? So our best estimate is we don't know anything biologically that would suggest that the vaccines would not work in MPS patients. Okay, so that's important. However, if we don't have any, to the best of my knowledge, if we don't have any immunogenicity data in that population, or we don't have any randomized controlled trial or observational data in that population, it's indirect. So we're extrapolating based on what we know and to the best of our knowledge, the vaccines would work to the same extent that they work in the other populations. So we know vaccine efficacy data from the Pfizer vaccine, from very large trials, from Moderna, and from AstraZeneca. So it is indirect, but to the best of our knowledge, we are, again, our best estimate is we feel that the efficacy would be in proportion to what's already been published in the general populations.
Perfect. Great, thank you. Okay, next question is, are there currently any vaccines available that are effective against the increasing COVID-19 UK, South African, and Brazilian variants?
So each of the vaccines, we're looking for data on that, and we have the more recent efficacy studies that have been published, where they were done during the time when these variants were there, have shown, for depending on the vaccine, some decreased efficacy against some of the variants, particularly the South African variant. There's less information from the vaccines that were did their efficacy studies first before those variants emerged. There's some in vitro data showing that the vaccines are still effective, but not field efficacy data. So that's a very much a changing area, and we're going to be learning a lot more about that as we start looking at effectiveness data, which is the real world data as the vaccines are used and the variants continue to circulate.
Perfect. Thank you. Okay, so my next question is, do you suspect any adverse events in like an immunocompromised or MPS population? If so, what are the probable scenarios?
Okay, we're going to switch around. You know, like in terms of adverse events, no. Just again, extrapolating based on data that's existing, right? So in terms of the adverse events, you know, at first people were judging the adverse events by the large clinical trials of, you know, roughly 30 or 40,000 people. Unfortunately, the numbers now have risen to hundreds of thousands, and there have been, particularly in the United States, very excellent surveillance systems that have not shown any particular, you know, worrisome adverse events. Of course, allergic reaction occurs, local reactions occur to the vaccine, but again, it's to the best of our knowledge, there's nothing suggestive that there would be any increase in adverse events in populations, in other populations. I could say that, and again, I've been thinking about this, whether this would be helpful for your association. I was part of the Canadian Rheumatologic Association panel for guidelines, because many, not most of patients with rheumatologic diseases are immunocompromised, right? And the question posed to the panel was, you know, very simply, should people with rheumatological diseases or illnesses receive the COVID vaccination or not? And it was a systematic approach, looking at all the evidence, looking at the antibody response, and ultimately it was a very thoughtful response, and it was a conditional recommendation. It wasn't a strong recommendation because everything was indirect, but some people wanted to make it a strong recommendation, but it ultimately was a conditional recommendation in support of it. And the panel, you know, essentially very much supported it, but just based on the way that these guidelines are made, they couldn't come up with a strong recommendation because you would need direct evidence or randomized controlled trial evidence, which is not present, which we don't have at baseline. So going through one of those exercises might be a consideration for your organization, maybe an international one or something like that. Just
Perfect.
a suggestion.
Yeah, even when I was doing my own research, like, I mean, there, as you guys both said, there isn't any data for immunocompromised populations. So, you know, a lot of the advice that the CDC is giving is, like, again, as you said, conditional. Like, you need to, like, it's really, we as our association and you guys as, you know, part of the task force, it's really up to your personal physician what they think, what they would advise, considering, you know, what medication you're on. Like, it's all just dependent on your circumstance.
Yeah, although I have to say the you know, again, I was very impressed by what the Canadian Rheumatological Association did, because although there was no COVID vaccine immunological data, they reviewed all the immunological data they could find in terms of vaccine related in their population, and sort of extrapolated from that. But it was a very, very thoughtful process, and it also included patients or patient representatives on the panel as well.
Perfect. Take that into consideration. All right. Oh yeah, Scott, go ahead.
No, I was just going to say, I think, you know, that for immunocompromised, I mean, for individuals with underlying diseases, you know, one looks at the vaccine, the platforms of the vaccines that are available, and doesn't appear to be an increased risk of using the vaccine. And from an immune standpoint, the only concern would be whether or not someone is going to have as good a response as an otherwise healthy individual. And if they don't have as good a response, does that mean they shouldn't get the vaccine? And I think most people would say no, if you know, unless there was no response, one would still potentially get benefit. And as Mark said, one wants to then say, well, with other vaccines, do we have any knowledge of the vaccine response being any different? Because again, vaccines are still using the same principles of the immune system of raising antibody and that antibody protecting. Now, there may be some T cell response too, but that's, you know, the main protection that we're seeing is likely through neutralizing antibody, and correlating that with other vaccines that have been used is a good principle that different groups, patient groups can look at to say, you know, what's the likelihood of there being any difference in our population.
Perfect. Okay, awesome.
Yeah, and if I can add one thing to what Scott said, is someone asked me this about a week ago about, you know, is there, you know, this sort of longitudinal symptoms in so-called long haulers in adults, is there data on children? And I wasn't aware of any. But I did a search, and there is a cross-sectional study that suggested, you know, a ongoing symptoms of 30 to 40%. Now, it wasn't the best study in the world, but to me, it sort of like says, well, you know, if people, even children, are going to linger on with symptoms for months, and a vaccine can prevent it, this is an important issue in terms of quality of life.
Perfect. Awesome. Okay, next question is, Pfizer recommends 2 doses within 21 days. What happens if you end up getting a delay in the second dose? Will it still work?
So I guess the issue is that in the studies that Pfizer did, that was the dosing schedule that was used, was 0 and 21 days. So that's where all their efficacy data is, and their approval was given based on that schedule. So from a company's standpoint, the company, you know, is going to say the vaccine needs to be used the way it was studied, because they can't advocate for its use in any other way. The likelihood is, though, that the vaccine is going to be effective whether it's given at 0 and 21 days, 0 and 28 days, because the immune system is still going to respond with a booster response well beyond that 21, 28 days or longer. So there will be data, real world data that comes on from observational studies where the immune response is measured in people who had longer intervals. So that one should try to keep to the schedule that it's recommended in the jurisdiction we're using, but it's not that, oh, if you don't get it at that dosage, you've wasted your time. That the vaccine is going to be working on all sorts of different schedules.
Okay, awesome. Thanks, Scott. Okay, next question is, what does the latest data show for transmission after getting either vaccine? So I assume, is the transmission like significantly lower? Do you still transmit the virus when you are vaccinated? Do you still shed? Like that?
Okay, so that's that's sort of an emerging topic, right? Because it relates to you know, what I just talked about herd immunity in this other meeting. And I would say that the data is emerging right now. So there is data from Israel that for the Pfizer vaccine, suggesting that you know, beyond I believe it was beyond 12 days, the viral load of those people who were infected after being vaccinated was lower than those who, you know, who were not vaccinated. Suggesting but not proving that might be, you know, they might be less likely to transmit. So the whole field of transmission is, I'd say it's a work in progress.
Awesome. All right. For the flu, people get their shot annually. Would it be the same for COVID? Will it be added to the flu shot eventually?
And that's an unknown. You know, the hope had been that it would be one shot or one series and done. But with the emergence of new variants, there may need, you know, depending on how the virus changes and whether the vaccines need to be changed, it may well require booster doses with different modified vaccines. We don't know that yet. And we also don't know the duration of protection. Obviously, these vaccines, even in the clinical trials, have just been used for under 6 months. So we don't know whether the protective immune response is 2 years yet, because the virus hasn't been around for 2 years. So that's all going to be information that we need to learn over the next 6 months to a year. And it's very possible we may need subsequent doses. Whether it's annual or every couple of years will really depend on the virus and how things emerge over time.
Okay, yeah. All right. Is the Moderna vaccine, quote unquote, more strong because it has a larger dose? What are your thoughts on that?
No, you know, first of all, the vaccines, you know, the answer is no. It's not a more strong. The question is of efficacy, and the efficacy of both Moderna and Pfizer vaccines are in the 90+%. So they are in the same, you know, the same ballpark in efficacy. If that was the question. Now, if the question of strength related to duration of protection over time in terms of comparative studies between vaccines, ideally, one would want to have a head-to-head comparison between two vaccines to be able to say this, and that data is not existing.
Okay. All right. A lot of questions are about specifically relating to lysosomal storage disorders, but we kind of established at the beginning of the Q&A session that we don't have data on that, and there has been no one with a LSD that has been in a clinical trial, so there's no known you know, results on that, so I'll have to admit a lot of those questions. So how are local health departments to find people with rare diseases and living with parents?
Can you say that again? How do?
Yeah. How are local health departments going to find people with rare diseases and living with parents?
I think, again, each jurisdiction will have different ways of identifying participants and or patients and or people, and asking them to, you know, come next. So as many different provinces as there are different ways of being rolled out. Some are doing it via their health cards and their health information that they have on hand, and are sending out invitations to be immunized based on their health cards. And that information has underlying illnesses and it has their addresses. Others are doing it, and some are doing by mail, others are doing it by text messages, others are doing it by advertising and, you know, electronic signing up. So that's really very jurisdictionally related to, because each province is rolling things out differently.
Okay, yeah, I figured. Okay, how is the efficacy determined? People aren't exposed to COVID-19 after, I guess, so how is the efficacy known? Maybe Rajiv, do you want to chime in on that one?
Yeah, sure. So basically, you provide the vaccine, and these people will go back to the communities. They will, be doing their own thing. When we designed the trial, we tried to recruit those people who have a more propensity to get exposed. So maybe the kind of work they do, maybe their work involves interacting with other individuals. So that sort of thing, just to increase the chances of getting those people who will potentially get infected at some point. So yeah, so basically you give the vaccine and the waiting game begins, because this trial was event-driven trial. So prior to the start of the trial, we calculated these are the number of cases we would need. We calculated the number of participants accordingly. And then after getting the vaccine, as I said, they went about their own lives. And at some point, if they contracted the diseases and developed the symptoms, then they will be confirmed with a PCR, and that's how a case is established.
Perfect. Thanks, Rajiv. Next question. So we only have around 4 more questions left. This one is, are there any specific considerations or recommendations for those who are post bone marrow transplant with vaccination?
Yeah, so bone marrow transplant is depending on how soon after your bone marrow transplant, very early after bone marrow transplant, there are the ability to respond to vaccinations is diminished. And typically, vaccinations aren't given within the first 6 months to a year after bone marrow transplant. And this is an area where an individual really should check with their bone marrow transplant team. And each center will have very specific recommendations on how they like to deal with not only COVID-19 vaccines, but all vaccines.
Thanks, Scott. Okay, so my next question is, if a child is under 12, will they be vaccinated at any point? Or are they expected to be protected by herd immunity?
Yeah, I can answer that. I mean, I think that the issue of herd immunity, first of all, is it's very hard to establish, right? So personally, I think herd immunity, you can develop herd immunity on a local level in a nursing home, in a school, but globally, my own view is that it'll be very difficult for herd immunity generally to be established, like smallpox. So it's good that what we want is we want both. We want people to be vaccinated. We want people to be directly vaccinated so they can protect themselves, and particularly even for children less than 12 years of age, there might be an ongoing risk again of there's less risk of severe complications, but there seems to be an emerging data of ongoing symptoms that occur. So I think that could be something. So I don't think that we expect people not to be vaccinated at all. But again, we want to wait for the data for some sort of, whether it's immunogenicity data or clinical data in those groups that weren't initially randomized.
Yeah, and the regulatory authorities are not going to allow the vaccines to be used in children until there's some data from children receiving the vaccine under clinical trials. And it may not be the large efficacy studies for children under 12. It may just be immunogenicity data. But those data, many of the companies are already planning on studies in children and pregnant women. And the likelihood is that that children will certainly be immunized, but it will probably be later on in the rollout.
Perfect. All right, the last 2 questions, and then we're done with this rapid fire. So, are there any reported interactions with meds? I mean, this question was specific to lysosomal storage disorder meds, but again, there was no data on that. So, just in general, are there any interactions with meds from this vaccine?
Most of the vaccines, again, there were people who were on what are called concomitant medications during the clinical trials. We haven't seen released any information about any analyses of people who were on medication. But again, unless a medication interferes with the immune response, there shouldn't be any problems with people on other medications.
Sweet. Okay, my last question is, for those who have been vaccinated, will they need a booster or a new vaccine, as they're saying it doesn't protect you from some variants?
Well, again, this is something that's ongoing, because it's a work in progress so far. So I would say personally, there's a good possibility that booster vaccines will be needed to protect against variants that are ongoing. But at this stage, there's no data to support that. So that's ongoing, and it's evolving with the epidemiology of the virus.
Perfect. So that's all the questions that were entered in the chat box. Kim, I'm not sure if you want to take over from here, but thanks for those responses, guys. That was awesome.
Yes, thank you very much. What I would like to do now, we've got about 15 minutes left for this webinar. If there are any questions now or comments that you'd like to ask the panelists or say to the panelists, please raise your hand and I'll open you up to the conversation. If not, what I will do is I will allow everyone to come into the conversation so we can have a discussion amongst ourselves. Dr. Halpern and Loeb and Rajiv, you're welcome to stay for this section as well. So I'll give you a couple of seconds to see if anyone's raising their hand. Not so far. So I am going to allow everyone into the room now.
Oh, Terry raised his hand.
Oh, Terry.
I did. Can you hear me now?
Yes.
Yeah.
Yes. No, I just want to say thank you to everybody for for putting this together, and also for the docs from Pfizer, and thank you for thank you for spending the time with us, us patients and parents of patients of MPS kids, to provide this very useful and very helpful information. Just thanks. That's all.
Thank you, Terry. We have somebody, Shane Watley, just asked, are there any fetal tissues used in the production or development of these vaccines?
Oh man.
God, do you want to go ahead, or does anyone have an answer for this?
So depending on the type of product, so the vaccines that are currently produced, I mean, I guess Rajiv can about the Pfizer vaccine, but for the mRNA vaccines, those are not made on cells, so there shouldn't be. And same with the AstraZeneca vaccine. It's made with a virus, as far as I'm aware.
Great. Julia Alton has raised her hand. Julia, please go ahead.
Hi, can you hear me okay?
Yes.
Hi, I just wanted to say thank you. This was an excellent webinar, so informative. I just had another question about herd immunity, and maybe you can help me understand. I'm confused if we don't know the transmission rate, or if we're not quite clear on how, if you get the vaccine and you get COVID, and you can still potentially pass the virus on to your family. How does herd immunity work then in this regards, if we don't really understand the transmission?
Well, I mean, herd immunity, the best way to study it is to actually vaccinate a group of people, and for whom the vaccine is licensed, and then see what happens in another group of people. So we've been doing these studies for many years for influenza, and whether we get to do it for COVID or not, I don't know. But that's really the only way you could determine that. So because first of all, a lot of the studies that are out there about shedding, whether people shed or not, the viral loads, first of all, it's a surrogate marker called a CT value, and it changes depending on the type of platform you're using, and how well that correlates to actually transmissibility is not really known, right? So, and for a lot of the vaccines, it's not clear how they're actually, are they causing something called sterilizing immunity, which means that the virus does not enter a cell and doesn't even replicate, right? Which would be ideal. Or does the virus enter the cell? Does it replicate? And is there the potential for shedding? But when you look at herd immunity, even if you have sterilizing immunity, even if you have, let's say, an antibody that from the vaccine that will go and hit that virus and the virus will never enter the cell, so it won't replicate, won't shed, we still don't really know the sort of threshold, the number of people or the percentage of people in a population that have to be vaccinated in order for the virus to stop. So we have theoretical numbers. So, in theory, it might be 60 or 70% for a perfect vaccine, or for 80%, close to 80% for a vaccine with 90% efficacy, but we don't have those numbers. So again, it's sort of a work in progress, basically.
Thank you.
Oh, and I should say that right now the safest thing to do is if someone is vaccinated, do not assume that because you're vaccinated, you cannot transmit the virus.
Okay. Thank you, Dr. Love. Thank you, Julia. That's a great question. I see Sandy and Jonas both have their microphones on. Were you wanting to ask a question?
No, I just want to say thank you. Okay. I think it was very, very informative, and there is a lot of unknown, but I think from what I heard, I think it's safe for my son to get it.
Great.
Glad to hear it.
Yeah, thanks, Jonas. And Sandy?
I know. I just wanted to say thank you to everyone as well. That was very helpful.
Okay, wonderful. Well, I see Julia. No, Julia, did you have another question? Your hand's still up. Nope, it's off now. Okay, well, wonderful. Well, we've got a little bit of time. So if anybody else has any comments, please speak now or forever hold your peace. Well, not necessarily forever. If there's anything that comes up that you think of after this webinar that you'd like to ask us, just email us either at info@mpssociety.ca, or you can email me directly at kimangel@mpssociety.ca. Kirsten Harkins has a question. So Kirsten, please go ahead.
Hi, Kim.
Hi.
You want me to, I put it in the chat, but I'll just put this forward. Thank you very much, everyone involved, for the really interesting series of presentations. I just had a comment because I was attending the World Symposium, that, as many of you know, a lysosomal storage disorder symposium. And one of the speakers there who was presenting on COVID and lysosomal storage disorders made one point that I noted. I have a son with MPS, an adult son, and this particular physician said that it would be best to avoid NSAIDs if you were being immunized because they may counteract the vaccine. And, I know a lot of people with MPS suffer from pain, and so may take Motrin, for example, regularly, or even sometimes during infusions. That's typical. So I just wonder if either one of the physicians could comment on that.
Sure, I can speak to that. There have been a couple of vaccines, not COVID vaccines, where it's been looked at to see whether NSAIDs or other anti-inflammatories or antipyretics or anti-pain medications have an effect. And there are a couple of vaccines where there has been an effect on the immune response, so that there's a little bit lower antibody levels. The meningococcal B vaccine is the one that's where there's most of the data that showed that. It's not been seen with all vaccines, and it was, even in that vaccine, it was only if the antipyretic or the NSAID was given prior to the immunization as a prophylactic. It did not occur if somebody was given the vaccine and then if they had a sore arm, an NSAID was used. So if somebody is not on NSAIDs and just uses them intermittently, the recommendation is for any vaccines, because we don't know, is not to use them prophylactically, but just wait. And if somebody has a sore arm or has a fever, then go ahead and use them. But there's no data at all with any of the COVID-19 vaccines.
Okay, thank you, Dr. Loeb. Good question, Kristen. Giannis, I see that your hand is up. Would you like to ask a question or make another comment? Nope. Okay, well, I think that's all the questions that we have today. Again, just our sincere gratitude for our panelists today. This was a very informative session. We've been fielding a lot of questions from our members and caregivers in the MPS community, and we opened this up not just to the MPS community, but to lysosomal storage diseases and the rare disease community as a whole. So I'm sure everyone here is walking away highly informed and grateful as well for your participation in this webinar. So thank you once again. And thank you to everyone who participated today. We hope to inform you of more of our webinars in the Education and Connection series that will be coming in the near future. And I will let everyone go 4 minutes early.
Thank you.
All right. Thank you very much. Yeah. Thank you.
Thank you.
Bye.
Thanks, everybody.
Thank you.
Thank you.
Everybody.